All right. Good morning, everyone. I see people are still jumping on, but I think in the interest of time, so we give Dr. Stewart his time to present. I think we should get started. So I'll turn it over to Dr. Bierkens to introduce him.
Hey, good morning, everyone, and welcome to Grand Rounds. We are joined today by Dr. Stewart, the director of the Diabetes, Obesity, Metabolism Institute at Mount Sinai. Dr. Stewart initially attended Trinity College and Columbia College of Physicians and Surgeons for medical school. He then was an internal medicine resident at Mount Sinai West and worked initially in private practice as an internist. Thereafter, he became an endocrinology fellow and later professor at Yale, prior to moving to Mount Sinai right here in 2012. His group was the first to identify PTHRP as the cause of humoral hypercalcemia. He purified and sequenced PTHRP and developed the current clinical immunoassays for PTHRP. At Mount Sinai, his team has described the first small molecule class of drugs that are able to induce human beta cells to regenerate for reversal of diabetes, which we'll hear about all today. This work has clear translational implications for type 1 as well as type 2 diabetes. We are incredibly excited to have him today. Please join me in giving him a very warm welcome. Dr. Stewart, take it away.
Thank you, Franz. Let me get a laser pointer here. Good. You guys can see me and hear me okay? Good. All right. So look, I'm going to take you on a little journey from, let me get rid of one more thing at the top of my screen. Well, I'll leave that. So a journey from impossible to possible, reversing diabetes through beta cell regeneration. I do have conflicts. Mount Sinai Tech Transfer Office has filed a number of patents on the work I'm about to present. So the first slide is simply a review of where we are in diabetes care, how effective we are. So this is from HEDIS. It's the scorecard, if you will, for how good we are as physicians in general in the U.S. at managing diabetes. So the goal is to get hemoglobin A1C under 7. That's the ADA, European, everybody's goal. Normal range is 4 to 6, but if we could just get to 7, that would be good. It turns out we're only doing that about 35% of the time. If you relax the standards a little bit and say, okay, well, let's just try and get to below 8. We're only getting there in about half of patients or less than half of patients. And if you say, look, let's just go for anything below 9, or how many people are actually above 9 hemoglobin A1Cs. It's actually astonishing. It's in the 40 to 50% range. So we're not doing very well in managing diabetes. So despite great drugs and years of progress, we still got a long way to go in controlling blood glucose in most people with type 2 diabetes. And the HEDIS data apply to type 1, but to type 2, but the results are not very different in type 1. So let's talk a little bit about the impact of diabetes. There are 37 million people in the United States right now with diabetes, mostly type 2. 96 million people have metabolic syndrome or impaired fasting glucose, or if you will, pre-diabetes. So that means 140 million people in the US, almost half the population, either have or are about to have diabetes. In the world, there are 437 million people with diabetes. So it's actually more prevalent in other countries outside the US in some cases. It's the number 2 or 3 cause of death in the United States, depending on how you count the numbers. And it's a major contributor to heart disease, stroke, and cancer, through which it causes death. It's the number 1 cause of, in the United States, of blindness, of kidney failure, of limb amputation, and through obesity and overnutrition, liver failure. 30% of people in the United States in hospitals are in hospitals because of diabetes, principally complications. And so that means that diabetes drives 30% of hospital expenses and revenues. The annual cost of diabetes, this is the most recent number from CDC, actually, from 2017, is $327 billion. Obviously, it's been going up as the prevalence of diabetes increases. And then finally, for families that have diabetes or for people that have diabetes, the cost is unmeasurable. What I'm going to tell you and what I want to focus on is that all diabetes is caused by inadequate numbers and function of insulin-producing pancreatic beta cells. So let's talk a little bit about pancreatic beta cells. We all have a pancreas. It's about the size of a strip steak. It sits up against the back of your spine. It's got about 1 million little pancreatic islets in them, and each islet looks like this. It's composed of about 1,000 cells, of which about 40% are beta cells. These are the cells that make insulin. You can take all of the islets in your pancreas, and it adds up to basically the amount, 1 cc, the terminal digit of any of your fingers. So it's a tiny amount of cells, but they're incredibly important. So the job of beta cells obviously is to continually sense glucose and secrete insulin. If your glucose goes up, you secrete insulin. If your glucose goes down, for example, if you're fasting or running, you need to turn off insulin. So what about beta cell mass in people with diabetes? Well, this slide is not surprising in one sense. We've known for about 100 years that in people with type 1 diabetes, beta cell mass is reduced. What's new here and publishable in this century is that this number is not zero. Everybody thought until recently that if you had longstanding type 1 diabetes with autoimmunity, all of your beta cells would be gone. It turns out at autopsy studies, and there are now lots of these, beta cells are not gone, even if you've had type 1 diabetes for 20 years, 50 years, 80 years, there are still beta cells remaining, at least a few. In people with new onset diabetes, the beta cell tank is only about 50 or 60% empty. So most people with new onset diabetes, type 1 diabetes, have residual beta cells in substantial numbers. So people think of type 1 as the beta cell deficiency disease, but it turns out that type 2 is as well. So again, these are all autopsy studies. You can't do imaging studies or quantitative beta cell mass studies in live human beings right now. So if you measure beta cell mass in type 2 diabetes here, abbreviated as TTDM, it's reduced compared to BMI, sex, and age-matched controls who are obese. So beta cell mass is reduced by about 60% in people who are obese. The surprise here again is that's not what people thought. People just thought this is about insulin resistance. Insulin resistance is an important contributor to type 2 diabetes, but if that were the only cause, then everybody who's obese would also have diabetes, and they don't. So type 2 diabetes is a disease of insulin resistance associated with a reduction of beta cell mass. So what this says is that basically everybody in the world needs more pancreatic beta cells. 537 people in the world, 1 million people in the world need more beta cells. So what do we do? Well, one approach is, look, let's just replace beta cells by doing a pancreas transplant. That turns out to be very effective. It's almost always done in the US and around the world in people with end-stage renal failure from type 1 diabetes or type 2. And so if you're going to transplant a kidney, why not transplant a pancreas from the same donor? That simultaneous pancreas kidney SPK model is actually super effective. The long-term survival of the kidney and pancreas grafts and the patients are extremely good. On the other hand, it's limited by the availability of organ donors, and it's major surgery. So there are only in the US right now about 90 of these being done per year, and almost all of them are done, excuse me, with kidney transplants. So what about pancreatic islet transplants? You can do that as well. You can take pancreases from organ donors, isolate the islets, get isolated islets like this, transfuse them or infuse them through a catheter that goes into the portal vein. This is done percutaneously in a radiology suite. And basically, the islets flow into the distribution of the portal vein, set up house in the liver. And they're also very effective. The issue here, of course, is there aren't enough pancreatic islets to go around and treat everybody. So again, in the United States last year, or in the last 5 or 10 years, there have been an average of about 70 of these done, and that's declining because of the availability of organ donors and regulatory issues surrounding approval of pancreatic islet transplant. So what about stem cell therapies? This lack of beta cells has fueled the stem cell if you will, wave to try and generate human pancreatic beta cells from stem cells that you could just transplant, and that would solve the limitations on the numbers of organ donors that you need for pancreatic transplant or pancreatic islet transplant. So the good news is this is amazing science. It works very well in animal models. And so far, it seems to be working well in one person who's had this. So the good news is this is amazing science. The bad news is it's only been done in a few people, and it's going to take a long time before this is scalable to millions of people with diabetes. So I'm a big fan of the science of this. I have a hard time seeing how this gets to 20 million people in the world with type 1, 500 million with type 2, and in the US, even a few million people. So, this is sort of the next obvious step then. Well, if everybody with diabetes needs more beta cells and they need it in a scalable way, why not come up with drugs that can make pancreatic beta cells regenerate? So that's a quest we set off on a number of years ago. And basically, this story all happened at Mount Sinai starting back in 2012. So what do we know about pancreatic beta cell regeneration? What we know is that beta cells can proliferate in human beings in the first year of life. So on the y-axis, we're talking about beta cell proliferation measured as Ki-67 and beta cells in organ donors and cadavers. So there's no beta cell proliferation going on in embryonic life. Your beta cells proliferate at about 2% per day during the first year of life, and then beta cell proliferation declines. And by the time you're an adult, even by the time you're a teenager, beta cell proliferation is essentially ceased, and no one's ever been able to come up with ways to make them replicate. So we set off on a high throughput chemical screen to try and identify drugs that would make beta cells replicate. I don't have time to go into all the details of how we did this, but a couple of points are important. First, the title says it all. We did a high throughput chemical screen to identify Harmine, which is an inhibitor of a kinase called DYRK1A, and this is able to induce human pancreatic beta cells to replicate. The work was driven by Peng Wang, who's shown in the picture here. Peng was with me in Pittsburgh before we came to Mount Sinai. And what we found is this molecule Harmine. And again, the bottom line is through a robotic screen of 100,000 molecules, we found this molecule Harmine, that's able to make human beta cells replicate. So what is Harmine? It's a small molecule. That's sort of med speak or chem speak for something you can take by mouth. Its target is a kinase called DYRK1A, and Harmine fits into the pocket in DYRK1A where ATP is supposed to fit. ATP is the phosphate donor for this kinase. So if Harmine is sitting in the pocket, then DYRK1A can't do its job. Its job is to maintain quiescence in beta cells. So here's what we did when we were 1 year old. Here's what we're doing now. Here's what Harmine is able to do. It's able to reproduce the rates of replication we all had when we were 6 months old. So the next thing is, well, that's pretty cool, but we're probably going to need faster rates than 2% per day to fill up the beta cell tank in people with type 1 and type 2. And also we worried that DYRK1A might, since it's expressed in every tissue, inhibiting that might cause problems. So we wanted to come up with ways to be more specific for beta cells. We just want to hit DYRK1A in beta cells. So we began to think about drugs that we use clinically, you all use clinically to treat people with diabetes that are beta cell specific, at least in a partial, in some cases, strong sense. So we use sulfonylureas, we use meglitinides, we use GLP-1 receptor agonists. All of these we use. And so we said, well, let's look at these and see if any of these will synergize with Harmine and give you more proliferation. It turns out the GLP-1 receptor agonist story worked. So this is this work of a wonderful graduate student named Courtney Kefi, shown here, later postdoc. And what Courtney showed together with Peng Wang is that GLP-1 receptor agonists synergize with DYRK1A inhibitors to potentiate functional human beta cell regeneration. What are GLP-1 receptor agonists? Well, I don't need to tell you all. They're basically widely used. The ones on the top are listed here are GLP-1 receptor agonists that are on the market or about to be. There's a small molecule GLP-1 receptor agonist. This is a paradigm for one. There are actually a lot in development right now. These are DPP-4 inhibitors, which achieve the same thing. They increase endogenous GLP-1 levels. So what's the relevance here? Well, they're in widespread use. 30 million people around the world are taking these already. They all work by increasing insulin secretion, but none of them induce beta cell replication. So when they were first discovered 30, 40 years ago, people were all excited. They might make human beta cells replicate. It turns out none of them do. So Courtney's experiment was this. This is her seminal experiment. She measured Ki-67, that's beta cell proliferation, in human islets from organ donors. And each of these lines represents pancreatic islets from a different human organ donor. She found that if you just put in vehicle, DMSO, there's no proliferation. If you added GLP-1, there's no proliferation. If you add Harmine, as we had shown earlier, as Peng had shown earlier, there's nice beta cell proliferation that averages 2 or 3%. The startling thing is that if you add GLP-1 and Harmine together, suddenly you're seeing much higher rates of human beta cell replication. So these are unprecedented rates of human beta cell replication. In the world of diabetes, this is really very exciting. What I showed you before is the effects of GLP-1, but what about all the drugs that are on the market? Exendin, exenatide, liraglutide, lixisenatide, semaglutide, none of them drive human beta cell replication, even though there are 30 million people are taking these right now. This is what Harmine does by itself, around 2.5% proliferation. In this slide, they're all normalized to Harmine at 1.0. But if you take any of these drugs and add them to Harmine, now you see, again, a dramatic and very reproducible increase in human beta cell proliferation. So this is extraordinary. This says that it's possible to make human beta cells regenerate at high levels. So we've gone from here's what we did when we were 1 year old, here's what we're doing now, here's what Harmine does. And if you add Harmine and GLP-1 together, now you get even higher rates of beta cell replication. And that's pretty promising for therapy in humans. So is this enough to treat type 1 diabetes? Well, 6 or 7% per day is pretty good. That should fill up this tank pretty quickly. In type 1, if you're starting here, it would take longer. But these are encouraging. So the next question becomes, well, I've been telling you that we have made beta cells proliferate. And our evidence for that is we're seeing pretty pictures of BRDU or Ki-67 labeling inside beta cells, inside cultured human islets. You could reasonably say, well, Andy, you didn't really show me beta cell proliferation. You showed me pretty pictures of islets with red nuclei, but you didn't show me that you got more beta cells. You didn't show me that you have more beta cells in the dish than when you started. So is the proliferation real? The technical issue here is that human pancreatic islets live in a dish for a matter of about a week before they sort of disintegrate and degenerate and stop making insulin. So trying to show increases in beta cell numbers in vitro was difficult. So we decided to use another model. And this is developed together with Adolfo Garcia Ocaña and Sari Stanley, also in the Diabetes Institute. So this is what human organ donor pancreatic islets look like. We get them on a regular basis from the NIH. We can transplant them into mice that have no immune system. We use RAG minus mice. So you can put human islets in these mice. You can put them in the kidney, under the kidney capsule here. And then you can withdraw the catheter, put the kidney back in the mouse. And now you have an immunodeficient mouse with human islets. And those islets will live in the kidney capsule for for the lifespan of the mouse, which is in the range of 18 months to 2 years. So now you can take these mice with human islets, you can put in little infusion pumps and continuously infuse Harmine or vehicle or exenatide or Harmine plus exenatide. And you can infuse them continuously for 3 months. And if you do that, at the end of a month, if you want to go longer, you can replace the pump. Right now, we've got out to 6 months, but I'm going to show you data out to 3 months. So at the end of the experiment, you can perfusion fix the mouse with anesthesia. You can harvest the kidney. The kidney is opaque. We need to clarify it for imaging. So we can use tissue clarity methods to clarify it. We can stain it for insulin so we can see the beta cells. And we end up with this clear kidney. And now you can do serial sections, if you will, using laser confocal light sheet microscopy. And you generate 3D images. So this is what these images look like. This is Adolfo. This is Sarah. So this is a mouse kidney with human pancreatic islets in it, and the green is showing the beta cells in the human pancreatic islets. This is an extraordinary video. No one's ever been able to see human beta cells in any living system. Obviously, this is formalin fixed, no longer living. But what it allows you to do is visualize the graphs. And more importantly, since it's 3D and you're measuring voxels now, you get from this technique human pancreatic beta cell volume. That's something no one's ever been able to assess in vivo in humans or in experimental studies in mice. And so the next thing is obvious. Let's take 100 islets, put it in a mouse and treat it with vehicle. Let's take another 100 islets from the same donor, put it in a mouse, and treat the mice with Harmine and exenatide for 3 months. And so without knowing anything about human beta cell biology or research, it's pretty obvious. Beta cell mass is increased dramatically in the animals treated with the combination of Harmine and exenatide over 3 months. You can quantify this easily now. Now we have voxels, so we know exactly the beta cell mass. If you transplant in 100 islets, take them out tomorrow or take them out the same day and study them, they all start off with the same number of islets. If you do an experiment for a week, at the end of a week, beta cell mass is down. That's because when you transplant human islets, they're ischemic, and many of them die in the first 24 hours. And so really the starting point is here. If you then look at 3 months, or sorry, at a month or 3 months, looking at vehicle or exenatide, beta cell mass doesn't change over 3 months. So you pretty much have at the end of 3 months what you put in on day 0 minus a few initial losses. On the other hand, if you treat with Harmine, beta cell mass increases by a factor of about 300%. And if you treat with Harmine plus exenatide, now beta cell mass increases by a factor of about 7-fold or 700%. So what I'm showing you is that you can actually increase human pancreatic beta cell mass by a factor of 7-fold, 700% over a period of 3 months by treating mice with Harmine, which is a small molecule, and exenatide, which is a avatar, if you will, for all of the GLP-1 receptor agonist drugs on the market, some of which now are available orally. How does it work? Well, I don't have time to go into all the details, but part of this is beta cell proliferation. Part of it is improving insulin production by existing beta cells. Part of it is enhanced beta cell survival and during transplant. We think that some of the beta cells are also being transdifferentiated from other, perhaps beta cell stem cells in the islets. And we think that combinations of these are happening. But remember this number, 700% increase in beta cell mass over 3 months. This is extraordinary. So if you're a person with type 2 diabetes and we were able to increase your beta cell mass by a factor of 7-fold in 3 months, where would you be? In 3 months, you'd be up here. That's like too much. So we'd have to use lower doses of these drugs or smaller doses of the drugs, for shorter duration. That would be obviously enough to fill up the beta cell tank in type 2. In type 1, it depends where you're starting. If you've had it for 50 years and your beta cell tank is down here, a 7-fold increase would get you up here, which would help a lot with beta cell stability. If you're with insulin stability and hypoglycemia, if you're starting off with your tank 50% full as new onset, this would be more than enough to fill up the tank. So these are remarkable events. We worried that if you make these beta cells proliferate, they're not going to work very well, right? So if you think about cancer biology, the most highly proliferative tumors are often the most dedifferentiated tumors. And so we thought if we're making beta cells replicate, maybe they're going to dedifferentiate and not work very well. So I won't spend a lot of time on this, but this is just looking at transcription factors and glucose transporters and insulin processing enzymes. And the idea is, we worried that these things would go down. These are all things that a beta cell must have in large amounts if it's going to be a successful beta cell. And we were looking to see if they would go down. To our astonishment, they all went up. So you can see insulin goes up by a factor of about 2 or so, about doubles. All these transcription factors go up. This is the glucose sensor that lets glucose get into beta cells. It goes up. This is an enzyme required for processing insulin. It goes up. And also glucose-stimulated insulin goes up as compared to controls. So you can have it both ways. You can make human beta cells proliferate, and you can also enhance human beta cell function. So this is, again, extraordinary, serendipitous, but wonderful news. What about treatment of diabetes? Well, we can do the same kind of experiment that I described earlier in immunodeficient mice transplanted with human islets. So we can take immunodeficient mice. These are RAG minus mice. We can treat them with streptozotocin, which you all know is to the extent that it's used anymore, kills beta cells in mice and humans. So if you treat mice with streptozotocin, you kill their beta cells. Now they become diabetic. Now you can put in a trivial number of human islets, let's say about 100, 150 islets. That's not enough to fix their diabetes. And then you can put in these pumps and infuse them for 3 months with Harmine, exenatide, Harmine plus exenatide, or vehicle, which in this case is water. So this is what these experiments look like. These are RAG minus mice, random glucoses. So blood sugars in mice are similar to humans. So this is postprandial. They've been up all night eating. So blood sugar is around 150. Give them streptozotocin, and their blood sugar becomes elevated. They have diabetes. Then you put in pumps. That's what these are, human islet. You put in transplanted islets and put in pumps, and then treat them with vehicle, and they remain diabetic. So if you just treat them with vehicle, they remain diabetic. If you treat them with Harmine and exenatide, shown here, blood sugar comes down, but it doesn't go to normal. On the other hand, if you treat them with Harmine plus exenatide, shown here, blood sugar comes down and stays in the normal range for 3 months. So this is amazing. This says that you can actually put in trivial, inadequate numbers of human islets. That's a surrogate for somebody with type 1 or type 2 diabetes. Got a few remaining islets. And you can treat them with Harmine and exenatide, and you can normalize their glucose completely for 3 months. There are 2 other parts of this that are not obvious from the figure. One is that at the end of the experiments, Adolfo harvested the kidneys and did the iDisco tissue volume measurement studies here. And again, these guys that got Harmine plus exenatide had about a 7-fold increase in human beta cell mass. So you can increase human beta cell mass not only in using normal islets and normal mice, but you can also do it you can increase it in a diabetes model. The other remarkable thing is, if I increased your beta cell mass 700%, that would be like having an insulinoma, and you'd get hypoglycemic. So what's pretty amazing and exciting is that these guys not only have reversed their diabetes, they don't get hypoglycemic, even though they have more than enough beta cell mass. So this says, that's good news for safety, but it's also good news for saying that these beta cells are functioning normally. They have the good sense to turn on when you need to make more insulin, and they have the good sense to turn off when you need to make less insulin. So again, this says that this should work well in humans. In these studies, Adolfo measured Harmine levels. Okay, so let's measure Harmine levels at different points during these studies in these various groups. I'm showing you data now from the end of the study, but it's the same for other parts of the study. So what these studies show is that animals infused with vehicle don't have any Harmine in their blood. Animals infused with exenatide have no Harmine in their blood. Animals infused with Harmine or Harmine plus exenatide do have Harmine in their blood. And the Harmine levels are around 5 nanograms per mil. So hang on to that number, 5 nanograms per mil, because I'm going to come back to it. 5 nanograms per mil is what you need to have on board to give you a 7-fold increase in beta cell mass, in human beta cell mass, and reverse diabetes. So what about safety? DYRK1A, as I mentioned, is expressed in many tissues throughout the body. So we were concerned that if we gave Harmine to normal volunteers, maybe other tissues would proliferate. So in the same studies that I've been telling you about with 3-month studies with Harmine and exenatide in mice, we harvested all the tissues. So we looked at liver, we looked at spleen, we looked at intestine, we looked at white adipose tissue, we looked at lung, heart, kidney, exocrine pancreas, and we looked at it in animals that got vehicle, that's what this little V is, and also in animals getting Harmine plus exenatide. And we measured proliferation rates, Ki-67. I don't have time to go through all of these, but just to make the point, here's liver. Beta cell proliferation rates are low in adult fully differentiated liver, and they don't change with Harmine or Harmine plus exenatide. In spleen, splenocytes are proliferating all the time in normal spleens, so they're much higher than liver, but they're no different in Harmine and exenatide versus the controls. So what this slide says is that in the very same animals that have a 7-fold increase in human beta cell mass and that's able to reverse their diabetes, there's no proliferation going on in other tissues, except for pancreatic beta cells, residual pancreatic beta cells in the mice. What about tissue histology? So we took these same organs, gave them in a double-blinded way to our veterinary pathologists: small intestine, spleen, heart, liver, kidney, pancreas, lung, adipose tissue. And not on this slide, it's a separate slide, brain tissue. And again, pathologists read them all as normal and said there's no difference between them in a double-blinded way. So it looks like 3 months of Harmine or Harmine plus exenatide is safe, has no adverse effects on tissue histology, has no off-target proliferation, and yet is able to reverse diabetes. So there's another safety issue to talk about. When we purified Harmine and published the Harmine story, we started getting emails from all over the world asking me if how much Harmine is in ayahuasca and if ayahuasca would cure people's diabetes. I had never heard of ayahuasca at that point, and so began to look into it a little bit. And so here's the ayahuasca story. There's a vine in Central and South America called Beta-stereopsis. There's also a plant called Psychotria viridis. And what the Mesoamericans 1,000 years ago, before the Incas, realized is you could take the bark of this vine and the leaves of this Psychotria plant, put them together in a pot, make an infusion, and you would have a religious experience or you would have a high. So that's been known for 1,000 years. Psychotria, by the way, is closely related to the coffee plant. So this is coffee down here. This is Psychotria. What's in these plants that gives them activity? Well, it turns out the Beta-stereopsis has Harmine and a couple of Harmine relatives, Harmaline and tetrahydroharmine, and these are monoamine oxidase inhibitors. Psychotria contains dimethyltryptamine. Dimethyltryptamine is a well-known hallucinogen. So it turns out that if you want to take dimethyltryptamine, DMT, and have a high, you have to snort it or you have to take it IV or sub-Q. Otherwise, it doesn't work. If you take it orally, it doesn't work. That's because in the GI tract and first pass in the liver, it's degraded by monoamine oxidases. It turns out serendipitously that Harmine is also a monoamine oxidase. So what the Mesoamericans realized, again, 1,000 years ago, is that if you took Harmine and DMT together, you could take it by mouth and you could have a nice high. Allegedly, this is restricted to the religious class, the priests. Okay, so what about now? Why are people emailing now saying, can you help me? Well, if you go onto Google and Google Ayahuasca Tourism or Ayahuasca Diabetes Tourism, you can find people that will take you out in the jungle in Brazil and help you identify the plants. You can find a shaman that will cook these things up and put them in a pot for you. You can find, you can either use a local shaman or you can bring your own shaman and his guitar from San Francisco. These are all on the internet. They'll put it in a pot, they'll cook it for you, they'll strain it, and this is what ayahuasca looks like. And then you can have a private party by yourself in the jungle. or you can bring all of your friends or patients with you to South America and have a group experience. So I'm being a little facetious, but actually this is all true. This is all real. These are all just downloaded from Google. So why is it so prevalent, and what's the story behind ayahuasca? Well, it turns out that ayahuasca is a central part of a religion in Brazil. The religion is called the União de Vegetal, which is basically the union of plants, and they believe that extracting plants is a way, and finding things like ayahuasca in them is a way to heal your spirit, heal your soul. And there are people in formal, well-recognized, established, state-approved religions in Brazil that use ayahuasca on a weekly basis. So you go to church, you have ayahuasca, and then you go home every Sunday for years, for decades. So this investigator, Calloway, who was a disciple of Timothy Leary, who's the guy that popularized LSD, decided in the 1990s to get down and investigate what really was in ayahuasca or ayahuasca, and what happened to people who take it. So he found 14 volunteers from this church, went to the jungle, got informed consent. The volunteers weighed about 70 kilos. The amount of harmine in the ayahuasca that their shaman made was about 250 milligrams. The amount of dimethyltryptamine, which the putative hallucinogen, is around 35 milligrams. And so he asked if he could just draw blood during this study, and he did. And these are the pharmacokinetic curves of harmine. So in people taking ayahuasca made under religious church supervision, had rapid increases in harmine levels in blood. It peaks at around 30 minutes, lasts about 2 hours, and then declines. So these are the peak levels of harmine in people routinely using ayahuasca. Dimethyltryptamine levels also increase. Remember, the dose of this was 250 milligrams. The dose of DMT was 35, so it's much lower. And then this is tetrahydroharmine, which is a degradation product of harmine. So it comes up later and lingers longer. So these are the physiological, or these are the routine pharmacological effects of ayahuasca on harmine. So remember this number, 90 nanograms per mil. People routinely using ayahuasca in religious services that have been going on for the hundreds of years and millennia are around 90 nanograms per mil. The values that it takes, circulating concentrations in mice with human islets to increase their human beta cell mass 700% is 5 nanograms per mil. So there's a lot more harmine in ayahuasca than is necessary to treat diabetes. This says the same thing, about 20-fold difference. So what do we know about human studies with harmine? Well, no one's really prepared human harmine under ways that we would consider FDA permissible these days, but there are studies back in the 50s and 60s where harmine was explored as a treatment for Parkinsonian tremor in people with Parkinson's disease, and also in people with drug-induced extrapyramidal tremors. And so these are the two studies that I've been able to find on this. They were done in less than ideal circumstances by current standards. There was no IRB, no informed consent. They don't really tell you where they got the harmine or how they made it, or that they're sure that it's really harmine. They didn't do any pharmacokinetics. They didn't really describe adverse events. They say that nobody died. And the highest dose of harmine that they were able to give is up to 960 milligrams. They stopped there, not because of adverse effects. That's just when they decided to stop. So they found no beneficial effects of harmine on Parkinsonian tremors or extrapyramidal effects of other psychiatric drugs. So they stopped using it, and that was sort of the end of it. So we said, you know what, it's time to actually look at harmine more carefully ourselves. Now that we know that harmine is so effective at making human beta cell mass expand, let's take a careful look at harmine in humans using pure harmine. In other words, let's do a phase 1 study. I see there's, okay. So Let me just stop for a second. I see that there's messages in the chat. Let me just see what those are about. Unfortunately. All right, so we'll come back to those at the end. Okay. Let me get the laser going again. Oh, got a pencil. Let me get a laser. All right. So it turns out that Mount Sinai has a wonderful clinical trials unit in psychiatry run by James Murrow. So there's a psychiatry clinical research center. We have been able to synthesize GMP, that means good medical practice, good manufacturing process, quality Harmine. We did that with the University of Minnesota that has a GMP facility. We've gotten approval from the FDA. We have our investigational new drug application approval. We have approval by Mount Sinai's IRB. We have a grant from NIH to do this study. And we've registered this trial at clinicaltrials.gov, which you must do with any trial. So it's a phase 1 trial. What are we trying to do? Well, we're trying to define how much Harmine a person can take without something bad happening. We have little in the way of guidance from prior human studies. I just described that. And so we don't really know what side effects to anticipate. And so this is sort of an open-ended study. Just push the dose until something bad happens. It sounds terrible, but in fact, this is done with every drug that's on the market. So you just push and try and find the maximum tolerable dose. We are allowed to enroll up to 40 subjects. They have to be healthy adults by FDA requirements. They're paid $350 to do it. They have 3 outpatient visits. They have a screening visit. Then they spend about a 10-hour day in the clinical research center. And then they come back 24 hours later for a follow-up visit. We measure blood pressure, heart rate, EKG, assess them for nausea, vomiting, diarrhea. Those have been associated with ayahuasca. Other symptoms, any other symptoms, we measure blood for Harmine and Harmine metabolites. These are normal volunteers, but we thought as long as we're doing it, let's measure insulin and glucose in these people. And they're also getting continuous psychological evaluations. This is important, right? I mean, one of the questions we're trying to ask is the psychoactivity derived from Harmine, derived from ayahuasca. Is that derived from Harmine or is that derived from dimethyltryptamine? I think most people think it comes from dimethyltryptamine, but there's never been a study with Harmine alone. So this is our chance to figure that out. This is what this stuff looks like. We've had it synthesized, GMP, University of Minnesota. You can see James Murrow's name on it, and Institute for Therapeutic Discovery and Development at the University of Minnesota. The pills are 100 milligram pills. This is the pill bottle for the first subject who was treated. And then there's a dose progression. So the first subjects get 100, and the next subjects get 200, then 300, then 500, then 700, et cetera, up to a maximum allowable dose of 1,200 milligrams. So where we are now is we're in the 300 to 500 range, and we haven't seen any severe adverse effects. A couple of people have gotten a little nauseated, but nothing bad is happening, and we're basically just trying to fill out the numbers here. What I can tell you, though, is that the levels of Harmine in the blood of these people and the doses are telling us that we've got a safety window of about 200-fold. We need to get levels of 5 nanograms per mil. We're about 200 times higher than that at doses that are well tolerated orally. So this is an ongoing project. Hopefully this gets done in the next 6 months or so. So people worry about using mitogenic drugs. If Harmine is a mitogenic drug, is that going to be bad? Well, we're seeing no safety signals saying it could be dangerous. And then we're reminded that people take estrogens for years, sometimes decades. People take testosterone for years, sometimes decades. Obviously, those are oncogenic if you do it at the wrong doses for the wrong amount of time. Growth hormones associated with colon cancer, parathyroid hormones associated with osteosarcomas. Insulin itself is really through work done here by Derek Leroyth, is associated with increased cancer risk in multiple organs. So here we're talking about using Harmine, which so far has no mitogenic signal, and we probably only have to give it for 3 to 6 months. So we think that's reassuring. Let's do a little benchmarking. How does this stack up against other things that people are using for diabetes? So what is our sort of current standard of care? Well, as I mentioned, we do pancreas and islet transplant right now. I gave them a 4 out of 4, in other words, a perfect scale for efficacy. Pancreas transplant works, particularly if you're using a kidney from the same donor. It's very successful. Long-term survival is great. Long-term survival of the graft is great. And it's life-changing for the person who receives a kidney and a pancreas. On the other hand, it's not very scalable. There are only a few of these done per year in the US. It's extremely expensive, and it's limited by the availability of organ donors. The cost per patient is extraordinary, extremely expensive, and it's not entirely safe. People on immunosuppressive drugs for organ transplant have about a 3% risk of getting lymphoma, lymphoproliferative disorder, or some other abnormality. And yet, is it in use? Yes. This is the only currently approved therapy for diabetes, for organ beta cell regeneration, beta cell replacement. Stem cells, I gave them a 3 on a scale of 4. 3 because they clearly work. If you take stem cells that are generated in laboratories, transplant them into mice with diabetes, just like I showed you with our work, they're very good at making mature beta cells. They're very good at reversing diabetes. I gave them a 3 instead of a 4 because so far it hasn't been shown to be useful on a large scale. There's one patient reported by Vertex who came off of insulin. I've heard that he's actually back on insulin now, and there are other patients who've 2 more in this trial, and they've not achieved insulin independence. So, is it amazing science? Yes. Is it going to be good? Yes, probably someday. Is it scalable to 500 million people in the world with diabetes? No. Is it scalable to 20 million people in the world with type 1? No. Is it going to be used in more than just a handful of people? It's unlikely. Cost per patient will be extraordinary, and stem cells have their own risks. I don't believe that teratoma is a huge risk, but that's there as a placeholder for other risks that we don't know about. Is it in use? No, it's not approved by the FDA. It's in its phase 3 trials now, if you will. So if you think about beta cell regenerative drugs from this perspective, is it effective? Yeah, the efficacy data that I showed you are pretty much identical to what the stem cell world has done with stem cell derived beta cells. So the beta cell regenerative drug story is that it works. Is it scalable? Absolutely. 30 million people are taking GLP-1 receptor agonist drugs right now. That's amazing. And obviously, if they had to add a Harmine pill to that, that would be trivial. Cost per patient would be trivial. Is it going to be oncogenic? We're seeing no oncogenic signals, but again, this will have to await large animal preclinical trials and then clinical trials. So what about autoimmunity and type 1 diabetes? That's still a big hurdle. It changed a couple of weeks ago. The FDA approved the first drug that's able to delay the onset of diabetes. This drug was actually described by Kevin Harold, who visited and gave grand rounds at Mount Sinai about a month ago. So it's a monoclonal antibody against a T-cell surface antigen called CD3. The name of the antibody is anti-CD3 or teplizumab, and it was used in a prevention study. They reduced the incidence of new onset diabetes by about 50% over a period of three to 5 years. So this is not a cure for autoimmunity. On the other hand, it's the first drug in 40 years of trying that actually delays the onset of diabetes. These studies are prevention studies because it's already been well established that once their beta cells are lost, giving autoimmune therapy, just putting people on prednisone for kidney transplant, is unable. It turns off autoimmunity, but it's unable to cause beta cell regeneration. So what this field needs is beta cell regenerative drugs. What we need is immunosuppressive or immunomodulatory drugs. So this field is now beginning to come together. Anti-CD3 is not the only one. This is another paper in the New England Journal that was reported recently, and there are others as well. Here's one in Lancet Diabetes and Endocrinology. So the autoimmune world is also making progress in parallel. So I'm going to end with a series of predictions. So I hope I've convinced you that human beta cell regenerative drug therapy is clearly possible. It'll be effective in tens of millions of people with type 2 initially, and then move to type 1 diabetes when immunological challenges are met. It'll be much cheaper and far more scalable than beta cell replacement therapies, which I don't think will ever be deployed on a multi-million person scale. It'll be at least as safe as other replacement therapies in current or future use. I'm referring to pancreas transplant or eyelid transplant and stem cell transplants. So what are our next challenges? Well, we need to finish this phase 1 trial. Hopefully by the middle of this year or the end of this year, we'll have finished that. We need to develop more potent and selective DYRK1A inhibitors. Harmine is a natural product, so actually it's not also protected by patents since we didn't discover it. But together with Bob DeVita, who's shown here, and his team, Kunal Kumar and Shalata Subaswang, they've made about 1,000 different DYRK1A inhibitors. Bob is the head of the Drug Development Institute at Mount Sinai. He was formerly the head of medicinal chemistry at Merck. And so basically, we have a large library of next generation intellectual property protected compounds that are more potent and more specific than Harmine, so that they're next in line. So we need to complete the phase 1, have more potent and selective DYRK1A inhibitors. That's the slide I just showed. We need now to do large-scale preclinical safety studies in rats and large animals, probably dogs. If there are adverse effects in tissues outside the beta cell, then we'll have to identify a perfect beta cell targeting molecule. There are lots of potential examples of things that might be useful there, but our data says that we don't really need a targeting animal. And then finally, in people with type 1, we'll have to deal with autoimmunity. So the story that I've just told you is sort of a paradigm for translational research. It starts at the bedside, taking care of patients with diabetes, understanding what their issues are, understanding our success and failure rates, which remain high. Then it involves going to the bench, thinking of ways to understand how you might make beta cells replicate, why they don't replicate, et cetera, do high throughput drug screens, and then find drugs and go right back to the patient. So we use human organ donors for our eyelid studies. We're in the middle of phase 1 studies. That'll end up going to phase 2 and phase 3, hopefully. And then goes right back to people again, because then you'll have to optimize its combination with other immunosuppressive drugs, et cetera, et cetera. So this is sort of the great Mandela of translational research, which for me is the best fun of being a physician scientist, being able to see the whole picture. It obviously involves teams of people. So I particularly want to acknowledge Peng Wang, who initially discovered Harmine in a high throughput screen, and is still with us at Mount Sinai. He's focused on mechanism of action stuff, which I didn't really show. Adolfo Garcia Ocaña is the person who's been behind all the transplant stuff that I mentioned. Courtney was a grad student postdoc whose work I showed you, discovering the synergy of Harmine and GLP-1 receptor agonists. And then Sarah Stanley is the brains and expertise behind the 3D movies that I was showing you. And Bob DeVita, Kunal are MedChem people who've designed all these amazing next generation DYRK1A inhibitors. So I'm going to stop there. And if I can figure out how to get out of this, yeah. So I'm going to stop there, and I'd be happy to answer questions.
Thank you, Andy, for an amazing talk. You are right, Andy. There are some questions that were started in the chat.
So let me just see if I can get to them.
Some you may have answered already, but let's see.
Yeah, let's see. Are Harmine and exenatide able to be manufactured at a reasonable price? Yeah, I mean, so in the case of the GLP-1 receptor agonists, the newest ones, the basically now semaglutide is available as a pill. Everybody in the world who's overweight seems to be wanting to take that. That's super expensive, like $1,500 a month. On the other hand, a lot of the older ones are off patent and could be made generically, so they can be cheap. The GLP-1 receptor agonists. Harmine is simple; it's just a pill, so that ought to be inexpensive as well. So to me, that's part of the appeal of this. You could spend millions of dollars on doing transplants on people, just a few people, and maybe it's going to work, maybe it's not, or you could spend trivial amounts on GLP-1 receptor agonist and Harmine. So certainly this would be a lot cheaper than stem cells or transplant approaches. What is the source of Harmine, and how do you produce it? So we make it synthetically. It's made synthetically by Bob DeVita's made it here at Mount Sinai. It's just medicinal chemistry, organic chemistry, but it also can be extracted from plants. This Banisteriopsis that I mentioned. This is the next one is John He. So mechanistically, how does Harmine synergize with GLP-1 receptor agonist? John, that's the $64,000 question right now. We have a grant to try and explore that. The short answer is we know that it requires. So GLP-1 receptor agonists work through adenyl cyclase. There's a GPCR for them. They activate adenyl cyclase, cyclic AMP. We know they activate protein kinase A, but how that's interacting with DYRK1A inhibition downstream, we don't know. And we're trying to understand that literally right now. We can mimic it if we silence DYRK1A with viruses and add cyclic AMP, we can, to human islets, we see the same thing as when we add the drug. So we know those are the two pathways involved, but how they merge at a deeper level, we're still trying to figure out. And then let's see, Jill Barron, is the Harmine that might be responsible for emesis in some people who, is it the Harmine that might be responsible for emesis in some people who ingest ayahuasca? The answer is, from existing literature, it's impossible to tell if that's Harmine or ayahuasca, but I can tell you from our studies now giving pure Harmine, it does at way super physiological, super therapeutic doses. It does cause nausea and vomiting, but we haven't seen diarrhea. And so again, the therapeutic window there looks like the doses that we're going to be required to treat diabetes are about 200 times lower than the doses that induce nausea and vomiting. And then somebody says, thank you guys for listening to this. This is pretty fun. And then Ben Chen, do the GLP-1 receptor agonists on their own increase beta cell mass in your studies? The answer is no. Beta cell mass does not increase with GLP-1 receptor agonists alone. Do psychiatrists anticipate that Harmine may have antidepressant activities to boot? Great question. So that's exactly the reason we're running this study on this is James Murrow's Depression Anxiety Center, where they do psychoactive drug research. And so what I can tell you is in this phase 1 trial that we're doing right now at doses that are more than enough to increase beta cell mass substantially, we're not seeing any effects on antidepressant kinds of drug effects. So we'll have to see that we're still not done with this trial. We have to do more, and we'll know more of that. And of course, that's a 1-day trial in normal people. So longer trials have to be done as well. Jill Barron, nice comment. Thank you. Josh Tannenbaum. So the mechanism of nausea from GLP-1 versus Harmine. I would say the answer to both is that no one really understands what the mechanisms are. And if you think about it, it's not hard to understand. You can't assess nausea in rodents or dogs. And rodents, I hear, don't vomit. So we don't know how nausea really works. There are two obvious possibilities. One is a local effect on the GI tract, and there's a lot talked about that in the GLP-1 literature. But it's also likely central. There are GLP-1 receptors in multiple parts of the brain in regions associated with nausea and vomiting. On the other hand, Harmine also has serotoninergic effects that could be inducing nausea directly in the gut. So we don't know that, and we'd love to figure that out. It's become sort of an academic question because we're way below the doses that cause nausea. And then finally, Bob DeVita, as I mentioned, has made hundreds of hundreds of DYRK1A inhibitors, including ones that that are unable to enter across the blood-brain barrier. So we think that nausea and vomiting at therapeutic doses, even with Harmine and certainly with the next-gen drugs, is not going to be an issue. Let's see.
We can do one more question, Andy.
Yeah. Do GLP does GLP-1 enhance the Harmine effect? I mean, absolutely in beta cell mass, but I think you're maybe asking about does GLP-1 enhance the psychoactive effects of Harmine or ayahuasca? The answer is I have no idea. And GIP. Oh, or maybe this question is, it says GLP and then later on it says GIP. So, this new drug that I mentioned, that's a combination of GLP-1 and GIP, this is the new Lilly drug for weight loss. That activates cyclic AMP inside beta cells. So we assume it acts the same. We actually have just gotten some of that. We're going to test it ourselves. So stay tuned for that. Happy to answer any other questions.
Dr. Stewart, I want to thank you on behalf of the department for coming and presenting today. It was an absolutely fantastic talk. I never expected us to be going to the middle of the Amazon after when you started talking about diabetes. So absolutely terrific, Andy, to watch your translational research here. So thank you very much.
Yeah, thanks. It's all Mount Sinai. We had the idea of doing this before we came to Mount Sinai, but we didn't have access to high throughput screening. And so part of me coming to Sinai was to do this, and it's turned out way better than anybody could have imagined.
Well, you proved it today. So thank you, Andy.
Thanks all. Thanks for your interest.
Have a great day, everybody. Thanks for coming. Happy holidays.